नमस्कार प्यारे शिक्षार्थियों, मैं डॉक्टर शिल्पा विनोदिया, असिस्टेंट प्रोफेसर डिपार्टमेंट ऑफ बॉटनी, पंडित सुंदरलाल शर्मा ओपन यूनिवर्सिटी छत्तीसगढ़ बिलासपुर आप सभी का स्वागत करती हूँ। आज का टॉपिक है हर्बेरियम टेक्निक्स। Herbarium are the storehouse of mounted dried and poisoned plant specimens on a mounting sheet meant for numerous scientific and social assistance. The herbarium sheet is actually voucher specimen. Sometimes we confuse that we say voucher specimen as a herbarium sheet और herbarium sheet को हम voucher specimen कह सकते हैं। Herbarium sheet preparation starts from the plant collection, leading to the final storage, preservation and utilization. Some special techniques like digitalization of herbariums, etc. The use of herbarium is related to a large number of fields of study, including morphology, taxonomy, ethnobotany, pharmacology, plant breeding, and physiology. Luca Ghini in 1556 is the sole initiator of the art of herbarium making, who started collecting, drying and pasting them over a paper which was in the form of book. But later in 18th century, Linnaeus started a new method in which he mounted his specimen on single sheet and started storing them flat horizontally, which is followed by almost all the museums and herbaria in the world. It involves 5 steps. First one is plant collection, second drying, third one is poisoning, fourth is mounting, fifth one is stitching, and the next is labeling and deposition. So let's start with the first point, plant collection. Plant collection material required for sampling are digital camera, newspaper, absorbent sheet, plant cutter, digger, polybags, field notebook, marker, ball pen, magnifying glass and first aid kit. Specimens of angiosperm, gymnosperm and pteridophytes can be collected with their reproductive parts, that is flower and fruit, and it must be collected with their reproductive parts. Field work is maintained in a small notebook in which date of collection, location. Field note में हम ऐसे पॉइंट्स को लिखते हैं जो कैरेक्टर्स बाद में डिसअपियर हो सकते हैं, जैसे coloration of foliage and floral parts, corolla venation, anther coloration, before and after dehiscence, pollinating agencies, texture of foliage and perianth part, color and nature of fleshy mature fruits, habitat and exact location. For that we use the proximate object near the site. Then waxed pattern of shoot and root system, whether it is present or not, insecticides or repellents are there or not, branching pattern of shoot and root system, and then types of soil, moisture content, slope and light condition, etc. Pressing and drying जो टॉपिक है, इसमें हम क्या करते हैं, कलेक्टेड प्लांट स्पेसिमेंस को हम पहले प्रेस करते हैं एब्जॉर्बेंट शीट में, एंड ऑल द फ्लोरल पार्ट्स को हम क्या करते हैं, स्प्रेड करते हैं केयरफुली विदाउट ओवरलैपिंग इन ओरिजिनल शेप। लॉन्ग स्पेसिमेंस को हम फोल्ड करते हैं V, M, N, L और Z के शेप में। हार्ड एंड ड्राई फ्रूट्स वर केप्ट इन अ बॉक्स विद द टैग including collection number, naphthalene balls so that it can be prevented from insects, were kept in herbarium cabinet with their voucher number. Then पॉइजनिंग जो स्टेप है, इसमें हम प्लांट की पॉइजनिंग करते हैं। हियर ड्राइड स्पेसिमेंस वर पॉइजनड बाय डिपिंग द स्पेसिमेन इन अ सॉल्यूशन ऑफ मर्क्यूरिक क्लोराइड इन इथाइल अल्कोहल dissolved in 4.5 liter of ethyl alcohol called Q mixture. After poisoning, the specimens were dried and affixed on the mounting sheet. It contained the following information: herbarium sheet or voucher specimen. The general size of the mounting board or herbarium sheet is 42 into 28 centimeter. The label with the size of 12 into 8 centimeter is generally pasted on the bottom side or right hand corner of the mounting board. The specimen is pasted as far as possible in the middle, field number at middle of left margin and accession number and or barcode at right top of the herbarium sheet. Then comes identification. प्रेसिंग करने के बाद में हम उसकी आइडेंटिफिकेशन कर सकते हैं. यूजुअली आइडेंटिफिकेशन इज कंसिडर्ड टू बी द प्रोसेस थ्रू विच अ स्पेसिमेन हुज नेम इज नॉट नोन इज रिकॉग्नाइज्ड बाय इट्स कैरेक्टर्स टू बी सिमिलर टू सम known plant and accordingly given a name. But it is now felt that since no two individuals plants are exactly identical, this process should not be called identification but determination. That is why the annotation slips are called and marked determinative slips. However, the word identification is now so universally employed that it clearly signifies the entire process. Arrangement and maintenance. After the identification process, the specimens are usually arranged in the herbarium according to the recognized system or classification. In many Indian herbaria, the order and numbering of families and genera is according to the Bentham and Hooker's genera Plantarum. In few European herbaria, the families are arranged according to the recent classification APG 3 angiospermic phylogenetic group, but nowadays we are considering APG 4. The herbarium sheets are to be arranged in wooden or steel almirah with pigeonhole. Unattended herbariums get lost because of these insects. So, to protect the sheet from fungal and insect pest is an important step. So, contact insecticides used in herbarium are cyanide gas, para-dichlorobenzene, which is called as PDB, DDT. Digestive poisonous are the salts of mercury and arsenic. Mercury chloride is the insecticide. Then you can see in the diagram labels for voucher specimen, which includes heading, then name of the institution in which the specimen deposited, then scientific name, genus, specific epithet, author or authors, then families, locality and latitude and longitude, habitat, date of collection, name of the collectors, determined by whom, and then remark. If any remark is there, you can write over there. Then comes role of herbarium. Why it is important to prepare herbarium. So we will talk about the role of herbarium in teaching. So herbarium serve as an aid in teaching botany to degree students and postgraduate students. It is difficult for any student to identify local plants without the help of a proper herbarium. Nowadays बहुत सारे ऐसे सॉफ्टवेयर्स हैं जिससे प्लांट को आइडेंटिफाई कर लिया जाता है, बट ऐसा होता है कि वो प्रॉपर आइडेंटिफाई नहीं कर पाते, लेकिन हर्बेरियम से हम उसको प्रॉपरली आइडेंटिफाई कर सकते हैं. Many specimens which the teacher would like to show to his students may not be available fresh at the time of giving the course. In such situations, available specimen in the herbarium serves the purpose. Second is the research. In research, herbarium, which was considered to be the concern and tool of the orthodox taxonomist, is totally unanimously believed to be an essential requirement for biosystematic research. For biosystematic studies, including population studies, the worker needs material for his taxon from far and wide, as it is not conveniently possible always to visit different areas of occurrence of the taxon. One has to largely fall back on resources of the herbaria. Then comes ethnobotanical researches. The herbarium have proved to be very valuable source of information. Many native uses of plants recorded on the herbarium sheet have never gone into published literature and therefore have never been subjected to scientific scrutiny. Herbaria also provide a meeting place for discussions and exchange of ideas among scientists far from and wide. Now let's see the major herbaria in India. So, the well-known herbaria is the Central National Herbarium located at Howrah, established in 1795 and comprises about 2 million species. This is the first herbaria in the country and one of the most important Asian herbaria. Second one is Forest Research Institute, Dehradun, contains approx 3,50,000 species. The National Botanical Garden, Lucknow, contain almost 2 lakh species. Blatter Herbarium, St. Xavier's College, Fort Bombay, contains 2 lakh species. And Botanical Survey of India has herbaria attached to their regional centers and unit in different parts of India. So, this was all about the herbarium techniques and herbaria. We will meet in next video.